Hi, this is Ajay Anand and welcome to Science Classes from excelup.com. In this video, you will learn about the basis of classification of animal kingdom. So, there are various criteria on which we classify the animal kingdom, and some important criteria are levels of organization, body symmetry, nature of coelom, and patterns of organ systems. Levels of organization. There are 4 levels, or you can say on this basis animals can be classified into 4 types. Number 1 is cellular level, number 2 is tissue level, number 3 is organ level, and number 4 is organ system level. In case of cellular level organization, there is no division of labor. So, all the cells, they are responsible for all the biological functions. So, every cell will carry out respiration, every cell will carry out digestion or excretion, and so on. Cellular level organization is seen in porifera or sponges. In case of tissue level organization, some sort of division of labor is there. So, there are different groups of cells and each group is having different responsibility. Okay. So, there will be one group which will be responsible for respiration, another group shall be responsible for digestion, a third group will be responsible for excretion. Tissue level organization is seen in Coelenterata and Ctenophora. After that comes the organ level organization. So, now tissues they have evolved into organs. So, there are different organs for different functions. So, there will be an organ for digestion, there will be another organ for respiration, a third organ for excretion. So, organ level of organization is seen in Platyhelminthes and Aschelminthes. After that, there is further evolution, and now we can see there are groups of organs. A particular group of organ makes an organ system. So, in an organ system, all the organs, they are responsible for different aspects of a particular function. Say, you can take the example of digestive system of human beings. So, in the human digestive system, there are different organs like stomach, intestine, liver, and so on. And different organs, they carry different functions, but all the functions, they are related to digestion. Okay. So, organ system level of organization is seen in Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata, and Chordata. Even in case of organ level and organ system level, there are different types. So, for example, the digestive system, it can be incomplete or complete. So, when there is a single opening in digestive system which serves the gateway for entry of food and exit of waste material, then this case is called incomplete digestive system. So, incomplete digestive system is seen in Platyhelminthes. If there are 2 openings, one opening for the entry of food and another opening for exit of waste materials, then we say that this is a complete digestive system. So, all the other animals, they have complete digestive system. Circulatory system, it can be open or it can be closed. When there are no blood vessels, blood is directly flown on the organs, then it is called an open circulatory system. Arthropods, they have open circulatory system. When there is a pumping organ and there are blood vessels like arteries, veins, and capillaries, then we call it a closed circulatory system, the way it is in human beings. So, we have arteries, veins, and blood capillaries. Now, let us talk about symmetry. So, on the basis of symmetry, animals they can be divided into three categories: asymmetrical, radial symmetry and bilateral symmetry. When the body of an animal cannot be divided into two equal halves, even if you cut it from any plane, then the body is called asymmetrical. Most of the sponges or poriferans they are asymmetrical. When the body of the animal can be cut into two equal halves from any plane through a central axis, then this is called radial symmetry. So, coelentrates, ctenophores and echinoderms, they show radial symmetry. So, this is an example. This is a hydra, coelentrate. So, it has a cylindrical body with tentacles all around. So, if you view it from the top, it will appear like circle. Okay. So, a circle, it can be divided into two equal halves from any plane, and that is what happens in case of coelentrates or the animals with radial symmetry. So, whether you cut it from this plane or through this plane, you can divide it into two equal halves. When the body of the animal can be divided into two equal halves through a single plane, then this condition is called bilateral symmetry, as is the case with human body. So, human body it can be divided into two equal halves from this plane. So, Annelida, Arthropoda, Hemichordata and Chordata, all of them they show bilateral symmetry. Germinal layers. Germinal layers are basically the term has come from the layers of embryo, and different layers of embryo they develop different organs, they rather germinate different organs. So, some organ it develops from mesoderm, some organ develops from endoderm. Okay. So, in case of body cavity or in case of germinal layers, there can be two layers or three layers. On this basis, the animals can be diploblastic or diploblastic and triploblastic or triploblastic. So, when there are only two layers, then the animal is called diploblastic. So, the outer layer is called ectoderm and the inner layer is called endoderm. In between, there is an undifferentiated layer, and this is called mesoglia. In case of triploblastic, there are three layers: ectoderm, mesoderm and endoderm. So, platyhelminthes to chordates, they are triploblastic, and the animals below these phyla, they are diploblastic. Segmentation. In some animals, the body is externally and internally divided into segments with serial repetition of at least some organs. Okay. So, this is seen in Annelida. So, the body of Annelida or say earthworm is divided into different segments. So, the segmentation is externally as well as internally, and there is a repetition of some organs. Say for example, nerve ganglions, they will be found in every segment. Heart-like organ, it will be found in every segment. Nephridia is present in every segment. And this phenomenon, in which the body is externally and internally divided into different segments with serial repetition of at least some organs, is called metamerism. You may have a question that the body of Arthropoda is also divided into three segments: head, thorax and abdomen. So, what is the difference? The difference is, in case of Arthropoda, the segmentation is external only, there is no internal segmentation. In case of Annelida, the segmentation is both external as well as internal. So, that is the difference. Then comes another factor, notochord. Notochord is a rod-like structure which is present on the dorsal side of the body, and it appears during embryonic development. Notochord is mesodermally derived, which means it is derived from the mesoderm. Okay. Later on, in some animals, notochord may persist. In most of the animals, it disappears and it is replaced by vertebral column. Say, for example, our vertebral column, it was a notochord during embryonic stage. Okay. So, those animals in which notochord is present, they are called chordates. Hemichordata and chordata, they are chordates. And remaining animals from porifera to echinodermata, they are non-chordates because notochord is absent in them. Then another factor comes coelom or body cavity. Body cavity may be present or it can be absent, and on this basis there are three categories of animals. Number one is coelomate, number two is pseudo-coelomate, and number three is acoelomate. Coelomate means that body cavity is present. Pseudo-coelomate means that false body cavity is there. It appears like a body cavity, but it's not exactly a body cavity. And acoelomate means that there is no body cavity. So, now let us see what is the difference. These are the diagrams of various layers. This is ectoderm, okay, and this is endoderm, and this is mesoderm. So, in case of coelomate, there is a cavity which is lined by mesoderm. Okay. So, organs, they develop in these cavities, right. So, Annelida, mollusca, arthropods, echinoderms, hemichordates and chordates, they are coelomates. Body cavity is present in these animals. In case of pseudo-coelomates, mesoderm is present as scattered pouches. There are different pouches, okay, instead of a coelom. So, these pouches, they appear like body cavity, but they are not exactly body cavities. And Aschelminthes, they have pseudo-coeloms. Platyhelminthes, they are acoelomate. So, body cavity is absent in Platyhelminthes. So, this is all about various criteria of classification of animal kingdom. Now, let us look at the broad classification of animal kingdom. So, the animal kingdom, there can be different levels of organization, cellular level organization, then tissue or organ level organization. Okay. In case of cellular level organization, animals, they are mostly asymmetrical and acoelomate. Example is porifera. Now, when it comes to tissue and organ level organization, there can be two types of symmetry, radial symmetry and bilateral symmetry. The animals with radial symmetry, they are acoelomate, body cavity is absent in them. Coelenterata and Ctenophora, they are examples of radial symmetry. In case of bilateral symmetry, now there are three types: acoelomate, pseudo-coelomate and coelomate. Platyhelminthes, they are acoelomate, body cavity is absent. Aschelminthes are pseudo-coelomate, so there is pseudo-coelom, false body cavity. There will be scattered pouches lined by mesoderm. Okay. In case of coelomate, body cavity is present, and examples are Annelida, Arthropoda, Mollusca, Echinodermata, Hemichordata and Chordata. Now, Echinodermata has been shown with a different color, and there is a region. In some of the Echinodermata, based on the stage of development, the symmetry can be radial, and in some others, the symmetry of the body can be bilateral. Okay. So, that is all about the basis of classification of animal kingdom. Thank you.